Alain, thank you very much for joining us. Would you like to tell us a bit about your application and what your research focus is?
So I work on mostly on brain development and also on human embryology. So I try to understand how organs are being built during development in human, but also in different species. I also work on the evolution of development from birds to fish to reptiles, etc. So I try to really understand how different organs are built, where and when the different cells that are in your organs are coming from, and how they migrate in the body. So basically, how you build a human body during development in an embryo.
Yeah, and you obviously make substantial use of 3D imaging, in particular light sheet microscopy. Why is that? What made you go for that decision to use?
Also, there are different. I mean, during development, the shape of bodies, whatever the species, is changing very quickly, and it's very difficult from section to understand how things change in 3 dimensions. And anything that was available, if you study human embryos, for instance, come from studies done until the 50s where they use very thin sections. And it's very difficult, especially for MD students also, to understand how things are evolving very quickly. You know, in a few days, it can change completely. In a mouse, in a few hours, the shape will change. So if you study it in 3D, then it's also much better to understand, to visualize things, processes. So it's really a plus. And also for rare tissue, like the one we used, if you have one 3D sample, then you get a lot of information from one sample. You don't need to have 10, cut them in different plane. With one only, you can already understand a lot about what happens.
Can you tell us one example of a discovery that was possible only with light sheet microscopy, something that was not known before?
I mean, there were... there's a lot. I mean, we have described using 3D imaging in particular, as I said, the development of many structures in the body that were not described before. So we did for like lacrimal gland in the eyes, where we had no description of it, the development of the tongue and the nerves. It's also something that was not known. Just the development of human arteries, the way they form in the brain and in the whole body. The data, they are coming from 1947, and they're not very precise, and there's a lot of mistakes. So we were even able to find some features of the human body in the embryo that when we checked, told us that what was known about human adult anatomy was also wrong. So it's sometimes, because you see the whole body at once, it gives you a lot of information. So again, I could list many things, and it's every day. I mean, often what we see the first time that someone has seen it. So it's really something that is interesting.
Yeah, that's great. When talking about 3D imaging, we must also discuss the sample preparation, because that's quite a challenging part. Could you guide us through your journey through 3D imaging, and how do you get to the level of imaging that you are now?
Yeah. So, I mean, to me, it's all these 3D things started, I know the day, you know, I was visiting a colleague from Bradke in Munich, and in his lab he had a student, Ali Ertürk, that I guess you know, works in Munich now as well. And Ali and Frank, they showed me a first movie of a mass spinal cord, but there are some axons in 3 dimensions. I'd never seen a 3D movie before. It was not published yet, and we're in 2010. And so when I came back to Paris, I said, I need to try to implement this in my lab. And that's when I got the first, also decided, when it became commercial, to get the first light sheet system built by Lavion Biotech with no Miltenyi Biotech, or part of Miltenyi Biotech. So basically, so it's a long, so we started 14 years ago. And of course, we have to keep improving the technique. The big challenge was to try to have a way to have antibodies working in 3 dimensions. So how can you get, in a thick organ, a big organ, have something to visualize cells penetrating deep into the tissue? And that took us a year to develop a good technique for it. So, and now we've been applying with more than around 2,000 antibodies. And the process we use is the same since 2014. It hasn't changed. So it's well established. It's people are publishing new improvement, but honestly, what we use is still, I think, is quite useful and powerful.
Yeah, from what I can see from your publication, it's clearly working still very well. You mentioned 3D antibodies. You've had the chance to test some of the Miltenyi 3D antibodies.
Yeah,
Can you
we
tell
really
us a bit more about that?
Yeah, that's to us that the main thing we're trying to find is we always need new antibodies. As I said, already tested more than 2,000 antibodies. So in my lab, that's the only thing that is like locked, in freezers with alarm on, because there's like thousands of euros of antibodies. And for sure, everybody is doing like immunostaining has the same problem. You're very happy with an antibody, it's great, and then, okay, there's no more of it because they are not recombinant. So that's one thing that is good with the military antibodies, that you make recombinant antibodies. So it means that normally you should always get the same quality over time. So we've been testing a lot of the conjugated ones. So it's also another thing we try to do multiple rounds of stainings, either in 2D or in 3D. And for this, you need to have conjugated antibodies. So it's also something that is good because Miltenyi has a lot of very good conjugated antibodies in the pipeline. So we've been testing a lot of them, and so to us, the penetration is much better with this type of antibodies, and the fact that a recombinant is such that normally we know that in 2 years from now, the quality will be the same. So, but there's always a need to get more. We don't have enough. So,
hopefully we'll be able to provide more and more.
And we got 20,000 genes, so you have maybe 40,000 proteins if you count the variants. So of course it's hard to have antibody for every specific target.
Yeah. Regarding the instrument, you mentioned that you started your early career already with ultramicroscopes. You now have multiple ultramicroscope Blaze systems. Can you tell us a bit more about that? How do you find them? If there's a clear advantage that you see?
Yeah, for sure. We see it evolve. As I said, we got the first ultramicroscope, and we also got the first Blaze. So in the end of 2019. So I know we have 3. So we've been following all the progress. So it was not always completely smooth, I must say, but it's okay. But now I think it reached a point where it's really working beautifully. And also the big thing for us was the change 1 year ago of the high-speed mode. And I must say that was really like a game changer for us, because I work on very large sample. So it used to take like a week to scan something that now takes half a day. And so it's really a plus for us. We have a drawback, which is that your data sets are getting huge. So now it's a matter of how you're going to deal with something that is like 6 terabytes or 4 terabytes. But I think now the system is really, for us, the speed is really a key thing. We can scan at higher magnification, and the whole process is much faster. So you can really if you work on, let's say, mouse brain, like many people, you can scan like 20 in a day or 30 in a day, which is really something I couldn't do before. So you save a lot of time and money, of course. So it's really good.
You mentioned the amount of data that one system can generate can be overwhelming, and you have more than one. So how do you deal with those data? What is your current pipeline?
No, I don't know anyone that has a perfect pipeline, and so unless you have plenty of money, it's going to be an issue. So we're planning to generate maybe 2 or 3 petabytes of images over the next 2 or 3 years. So which is getting really problematic in terms of cost. So we're trying to, for now, we store them locally and also on some cloud system that have a cost. Well, now it's maintained, but the quantity of data also with other systems like the Maxima as well generate nice data that are very big as well. So it's really becoming a big problem. So we're discussing with different either companies or other solutions for compression of data. So it seems that you can, even with like a 7 times compression, you can still do deep learning, AI with the data. It doesn't change anything. So for the raw images, that will be the way we go, compressing them at least a factor like 7 times. But even this also have the cost you have to pay for a solution, etc. So it's an issue, and because people normally we're supposed to keep data, raw images in France it's 10 years.
Yeah.
So think of it, if I generate 10 petabytes of images over 10 years. So consider the cost of the cloud system now, it's going to be really a lot of money for my lab. But we have unique data. No one has was human embryo data or was real species data. So that's why we think it's really important to find a solution. So it's taking a lot of time trying to find the best, and you need multiple copies. Plus you work on your data with multiple solutions. Each time you double, triple the original size of what you have. So that's also something a lot of people that are starting to do clearing, they don't think about it. So they have the system and they neglect the fact that they're going to generate like terabytes of images. So, and that's sometimes what is blocking them in the process. So.
Yeah. So you clearly see this being the current bottleneck of the workflow.
Yeah, it's really one of the problems. The other bottleneck is that we've been doing in Paris for the clearing part. We did for many years tissue clearing courses. So we train 450 people from all over the world. So for like 4 or 5 years, they can learn very quickly. So they can get images after 2 days. They know to do it. The problem now is that they don't know how to analyze the data in 3D. That's where like the problem is for most people now. It's hard for them to make nice images. They don't know how to deal with it because it's big. They don't have the software that are good enough. They don't have powerful workstations, so they don't know what to do. So one thing we're trying to do more and more is also to work with like cloud system, like I can mention NVIDIA, for instance. You can have software running on a remote server. Then you need to have any kind of specific computer in your lab. You can just use the GPU that are provided by the supplier. So I think that will be the thing that people will do in the future because it's we're lucky to have many workstations that are each of them around 30,000 K, but it's not something that everybody can afford. So it's a problem because you get your data very quickly now. But then in the institute, if you have just like 2 or 3 workstations, then you're going to have to wait to be able to analyze your data for like a month maybe. So then there's no need to go faster if you just are blocked then for the analysis. So I think for many labs, that's the thing. They don't know how to analyze them and they don't have the workstation that are powerful enough. So they should think about it when they apply for funding. They should also include this part because the storage, the analysis is really a huge cost.
Yeah, absolutely. And we've been focusing mostly on data storage, but what about analysis of the data? You're also on the forefront on that side. You are using VR software, AI. Can you elaborate a bit on that?
Yeah, we use anything we can. I mean, it's just like there's not a perfect solution, but for sure again, what is frustrating, a lot of people, so there are 3D data sets, so 3D, okay? And then what you do, you open them, you visualize them in a screen that is 2D, okay? So it's very frustrating. That's why more and more, a few years ago, we started to work with companies that are using virtual reality. So which means that we can navigate in our data in 3D. So if I wear the headset, I'm inside the sample. I can look up, down, I will see it, I can move it, it's very fluid. You can also open 20 terabyte data sets with the software we're using. So now if you come to my lab, you will see every day like 10 people wearing headsets and analyzing things. And even to visualize, it's really something that is a big plus. We're also trying to work with other companies that has developed a beautiful display for 3D, where you don't need to wear a headset, you sit in front of it and you see your data in 3 dimensions just in front of you. And that's really impressive. I mean, it's for medical teaching and for many things. Even to display your results, you just sit in front and you see it. So there's a lot of now, there are other 3D data, more companies and more solutions are coming for the visualization. So you need to have different types of software, more and more as well. So the 3D data that we have, they're very big. And we want to understand things in 3D. So then you need to apply more and more like deep learning, AI solution, because it's huge. We're doing a project on the development of the human lung. And we have to understand how the branches in a lung where you have, 200,000 branches, we want to extract the length, the angle of bifurcation, the diameter for those 200,000 branches. So you cannot do it by hand, you cannot use VR, then you have to apply deep learning techniques. So it's really now, but we have good data sets, a lot of modelers and mathematicians that are working with us to help us because they're happy to have this type of data that didn't exist before.
2 questions on future outlook. Where do you see the field moving within the next 5 to 10 years? And if there's one thing you could wish for that it's not there, it could be a feature of the microscope, but it could be anything related to your research. What would that be?
In terms of development, I mean, multiplexing in 3D will be an issue. Like for now we use 4 antibodies, but we started to have some pilot protocols where we can do 3 or 4 rounds of staining, so we can go maybe all the way to 16 antibodies, but we like to be able to like the maximum, maybe 120 antibodies in 3D. So I think that's one thing that will be important to try to visualize as many cell types as possible. And the other thing, of course, is to be able to do like a 3D transcriptomics in 3 dimensions. So there are solutions, we're doing it like using HCR with different techniques, but it's working like with 4 probes or 4 genes at a time. And we like to be able maybe to do 50, 20, combine it with the immuno. So I think there's still a lot to do in terms of development technologically, and also for application for large tissue. You can image large things, but if you want to image like an adult human brain, there's no way you can do it with the existing system. I don't know if it's going to be feasible at some point, but that's the type of thing. But not everybody wants to do that. So I think So there are ways for improvement, but compared to when we started 14 years ago, there's still a lot of things that were improved, and like every 2 or 3 years, there's a new development, a new protocol. And also there are many more labs that are working on it. Initially, I mean, people didn't believe 3D would be very interesting. So for many years, there were not many other companies and other development of softwares. And now that they see there's really like a market and that it's working beautifully, then there are more and more new solutions coming and improvement, like in terms of image analysis as well. So there's still room for improvement on many aspects.
Yeah. Well, Alain, thank you very much for your time, and looking forward to hear your talk.
Thank you.
And see where your research goes in the future.
Thanks a lot.